Thanks everyone for coming along. Today I'm going to be talking about some spatial patterns and drivers of clonal richness in tropical seagrass meadows, and this has been part of a collaborative effort over the last 3 or 4 years. So just getting right into it. So we all know seagrasses are clonal plants. Their populations are maintained through vegetative growth and through recruitment from seeds. But the level of clonality that you find in a meadow, so the number of unique genetic individuals, is really dependent on the biological characteristics of the species. So their growth rates, their reproductive outputs and their successful recruitment, how these interact with the environment, and also the evolutionary history. So you often find on the range edge the genetic diversity and sometimes the clonal diversity will be lower, usually due to recruitment limitations. So this level of clonality has been shown to have really important implications for resilience and hence conservation and management. Studies in many terrestrial plants and in seagrass species are starting to show that where you have a higher clonal diversity, you have a greater resistance to pressures and disturbance. When you have a higher clonal diversity, you have more diversity in response, which provides, once again, more resilience to pressures. And where you have a higher clonal diversity, there's a greater probability of sexual reproduction and hence the potential for recovery when large-scale events occur. So the environmental drivers of clonality, it's been found that often stressful environments can influence the clonality of a meadow, so the number of unique genetic individuals in a meadow. And this is based mostly on terrestrial literature, where plants growing in very cold environments tend to have a lower clonal diversity, in nutrient-poor environments. And there has been some work in seagrasses that has shown in the presence of aquaculture, the clonal richness is a lot higher. So it's not only the stress in the environment, but disturbance can have profound impacts, particularly when it affects the recruitment rate of new individuals into the population or the mortality rate of those individuals within the population that you're studying. And a very long time ago, I love this model, Seagrasses and Thorne came up with this model for how disturbance rate relates to clonality. So they have a disturbance rate here along the bottom from low up to high disturbance rate and the number of clones in the meadow, which is similar to the clonal richness, so the number of unique individuals in the meadow. So when you have a really high disturbance rate, their modelling and some experimental work as well showed that there tended to be a low clonal richness. And they attributed this to mass mortality of genets, so a high mortality rate and very low recruitment. Whereas when you look at this moderate level of disturbance, they predicted higher clonality. And this is because the disturbance can facilitate recruitment and the mortality rate is not as high. But when you get to the situation where there's quite low disturbance, two scenarios or two sort of extremes can emerge. You can either have really low clonal diversity, and this is because there could be low recruitment and competitive exclusion, so the genets interact with each other and only one or two particular genets survive and take over. Or the opposite case is that you could have quite high clonal diversity, so even with just regular recruitment, even if it's very low, you can have a high clonality. And particularly if you have long-lived individuals, you can maintain that high clonal richness. So with that background, what I was really interested to look at was to understand the patterns and the drivers, so the spatial patterns of clonality using the measure of clonal richness, approximately the number of genotypes in a meadow, across a regional scale. So this study region was in the northwest of Australia, from Broome down to the World Heritage Area that you'll hear about a bit later from Simone, Shark Bay. It was over 8 degrees of latitude, and we sampled 2 species of seagrass, Halophila ovalis and Halodule uninervis, which are 2 colonising species. So for these two species, we used slightly different approaches for the population genetic markers. We used single nucleotide polymorphisms for Halodule uninervis, and we had 75 loci. And for Halophila ovalis, we used microsatellite markers, and we had 7 loci. With these types of markers, we had a really high probability of identifying the individual genotypes within the meadow, and we were likely to detect less than 1 in the meadow. So we were really happy with the resolution of the markers to identify the unique individuals. And we sampled 19 populations of Halodule and 14 of Halophila ovalis. We were trying to sample them in the same locations. It wasn't always possible because they didn't always grow together. Many of these were large boat trips where we'd end up on an island and we'd have to quickly try and find seagrass in a couple of days and sample and then move on. But we ended up with 12 sites where both species were present. So what we found was quite a lot of variation in the clonal richness in these meadows that we sampled. So for Halodule uninervis, at each site that we went to, we collected between approximately 30 to 50 samples. And when we did the genetics and looked at the genotypes in the meadows, we detected from 1 in one meadow up to 32. Halophila ovalis, similar sorts of patterns. So there was quite a variation in the clonal richness from 0 up to 0.72. So 0 means there was only 1 genotype that we detected in the meadow, up to 0.72. And in Halophila, it was a slightly higher range. There were some sites where pretty much every sample that we collected was a unique genotype. So there's quite a bit of variation there in that clonal richness. And what we found really interesting was when we plotted the sites where we had both species present, so this is Halodule and this is Halophila, there was a really strong correlation in clonal diversity. So the similar sites had similar clonal diversity for both species. So that indicated there might be some similar drivers there across those two species, environmental drivers that are affecting the clonal diversity. So what we did is for each site where we had both species sampled, we tried to, we developed some measures at that site for dugong grazing, disturbance from cyclones, which are really strong disturbance factor in the region, and some environmental variables such as temperature and turbidity that could be stressors for the species. And we also included an isolation measure. So populations that were quite isolated maybe on outer islands might have more recruitment limitation, which might, or less connectivity between other meadows, which could potentially impact clonal diversity. So for the dugong grazing, there wasn't one data set that covered the whole area, so we got access to all of the dugong surveys that we're aware of in the region, and from that, with the authors identified hotspots. So some sites were known for dugongs to occur and others were not. For cyclone disturbance, we worked with Mari Puutunen from the Australian Institute of Marine Science, who's a cyclone modeller, and what she developed was a probability that a site was exposed to extreme cyclone disturbance, and this is when they'd be predicted to be greater than 4 metre seas for more than an hour, and this was the annual probability over 15 years. So we predicted that these really extreme cyclones could rip up the seagrass and also potentially move the seed banks on. And the temperature and turbidity were extracted from MODIS remote sensing imagery by Ryan Lowe from UWA. So going straight into the results, we did a general additive, a GAMS model, a mixed model, and essentially we used the AIC, the IKK information criteria, to work out which environmental factors best predicted the patterns of clonal diversity. For Halodule, both cyclones and dugong grazing plus sea surface temperature were the best predictors of clonal diversity. Very similar patterns for Halophila as well. Dugongs, cyclones, and dugong plus sea surface temperature. So when we actually looked at the patterns, what we found with where there was high probability of cyclones, there was low clonal diversity. When there was a moderate probability of cyclones, it was a high clonal diversity. In the presence of grazing, it was a higher clonal richness. And increase in temperature in the presence of grazing resulted in higher clonal richness. So what could be going on there? I think with high cyclones, it can remove the seagrass meadows, the seed banks, and if this occurs over multiple years, which is the case in this region, it could limit recovery, resulting over time in this low clonal richness. Whereas where you have a moderate probability of cyclone disturbance, it's generating that opportunity for recruitment, but not bringing down the seed bank and limiting that recovery. In this part of the world, dugongs tend to be in very small herds, so it's unlikely that their feeding could remove genets and increase the mortality, but these small disturbances could facilitate recruitment. And you also get this positive feedback where fragmentation of the rhizome and grazing by dugongs stimulates flowering and can then provide more opportunities for recruitment and new genets entering the population. And finally, this interaction with temperature was really interesting. So if you look over a latitudinal gradient, often you'll have more flowering events or higher intensities of flowering in warmer environments and a reduced flowering period in the cooler environment. So this higher temperature could be stimulating more flowering that with grazing leads to more recruitment. So in this study, we identified that disturbance has really important implications for clonality in this region and temperature as well. And what we're really interested in moving forward with this is just understanding how stable this clonal diversity is. Over time, are these types of clonalities maintained? Do we see these types of patterns in other regions, particularly in this cyclone belt in the Indo-Pacific? What about other species? Would we expect disturbance to impact other species, say more persistent species that may be more resistant? And the other thing that I'm really keen to follow up on is this quite a number of meadows with really low clonal diversity, and what are the implications for this for resilience, and how can we incorporate this into conservation and management? Thank you. Gary.
Interestingly, you've got something on clonality. I suppose with high clonality, how did you find a lot of connectivity across that range, or have you looked at connectivity?
Yes, we have looked at connectivity of the populations where we have enough individuals to actually assess population connectivity. Half of our sites were sort of less than 10 individuals, which makes it really, you can't trust the connectivity estimates. So where we do have adequate data to do that, we're seeing reasonable levels of connectivity over 5 kilometres. And we're seeing some signals of some long distance connectivity which may be facilitated by dugongs, but it seems to be quite rare. But as you're aware, trying to understand the timescale over which that connectivity has occurred, we're not quite on top of that yet, and that's more an area that we need to understand better.